All right, here's just a quick answer key for the transcription and translation worksheet that I asked you guys to do before. I just want to double check that you guys are understanding what we're doing. And so in this activity, you were given either a piece of DNA, messenger RNA, tRNA, or an amino acid, and you had to try to figure out the missing strands. And so in this case, in number 1, if you look at this messenger RNA strand right here, they started with AUG. That's the code, or in this case, that triple letter codon we talked about. And so if we first work our way backwards, in DNA, A pairs with T, U would pair with A, and G with C. And so there's our code for DNA, and you continue this through all of these different codons to go back to the DNA. A pairs with T, C with G, and U with A, and you keep going on. And so if you want to just kind of pause and check to see, excuse me, that you got that right, then once you have your DNA strand, we go to the tRNA. A pairs with U this time because we're in RNA and not DNA. U pairs with A and G with C. And so you just go through and match the base pairs together. So you should have ended up with this for your tRNA line. And then remember, the last part asks you to look for the different amino acids that this is coding for. To do that, you have to go back to your messenger RNA strand, and you're going to get your codon wheel and look up AUG. And so when you look at that codon wheel or that genetic code, start with A, then go to U, and then go to G. It should bring you to the amino acid methionine, which is our start signal. Then go to ACU, and that brings you to threonine, and we just keep going using that codon wheel to figure out what each of these codons is coding for. And in the end, you get to UAG, and that should bring you to your stop signal. If this was your body trying to make a specific protein, it has a start signal, it has a stop signal, and if all of these were coded correctly, then your body would be healthy and it would be able to perform a certain task. Okay, so if we go to the second example, I'm just going to kind of click through each one, and you can stop and check to make sure that you did them correctly. Every single one of these examples, like in your body, should start with the messenger RNA code, AUG, which will always be our start signal. And then you continue one by one to get your amino acid sequence correct and get your stop codon, and you should be set. The third example, you have to work your way backwards and then find your amino acids. Excuse me, I hope I'm not getting sick. And then finally, our fourth one, this is the one that I wanted to go over with you guys. This one can be a little bit tricky because you're looking at the amino acids on your codon wheel. And in some instances, if you remember when we were talking about mRNA, some amino acids can have more than one codon for them. So what I have up here may not be what you have, and that's 100% okay. Methionine is going to be AUG right here. But then if our amino acid was valine, then we'd get GUG. Oops, I went backwards. Sorry, guys. Methionine, we would actually get our mRNA strand first and then go to DNA and tRNA. So to get methionine, you go to AUG. Arginine, this would be one option. And for some of these, there's only one option, but for some of them, there's a couple of different ones. And so yours might not match up, and this would, number 4 would be the only one where it doesn't match up correctly with mine. And if you were to get a question like this later, you just pick any one of the number of possibilities for a code that matches up with some of these amino acids, and you should be set. Otherwise, the other questions that you had to answer were more open-ended. What are the 3 differences between RNA and DNA? Hopefully you can look back at the video or your notes. One of the differences is the sugar. RNA has the sugar ribose, where DNA has the sugar deoxyribose. RNA is smaller, it's a single strand, where DNA is double-stranded, and then the differences here left over are the bases, where uracil replaces thymine that was found in DNA, and uracil is found in RNA. So where is DNA found in eukaryotic cells, like what we have in our body? That should be an easy one for you, the nucleus. And what are the different strands of RNA molecules? Messenger RNA can be found in the nucleus where it starts, then it brings that code out to the cytoplasm and parks itself on the ribosome. So there's a few different options. Ribosomal RNA can be found in the cytoplasm, and transfer RNA is also going to be in the cytoplasm and also makes contact with the ribosome when it brings in its anticodon and the amino acids. So the three strands of RNA, what do they do? The messenger RNA decodes that information on DNA, so it transcribes it or rewrites it to a different form that it can understand. rRNA helps in translation. It provides a place where the messenger RNA can come, and then it reads that code on mRNA. And then tRNA brings in the anticodon that matches the codon on messenger RNA, and it brings in a specific amino acid to match that to make a protein. It says to draw a messenger RNA strand that would be complementary to DNA, you're just looking at these base pairs here and making sure you know what each one pairs with. It should be UUAACG. And then the smallest component of these strands would be a nucleotide, and we just want you to understand the three parts that make up a nucleotide. The phosphoric acid, the sugar right here, and this would be the rungs of that twisted ladder, the base. So what are the steps of transcription? If you look back in your notes, there's a few different things that happen. RNA polymerase has to unzip DNA, and then it has to eventually also bring in the correct nucleotides so that it can read that code that's found on DNA. So once it finds that promoter site, kind of like a start signal to start working, it unzips it and brings in the nucleotides, and then it goes all the way until it has a stop signal. And then it takes that code and leaves the nucleus, and that's when translation starts to take place. That messenger RNA lands on the ribosome in the cytoplasm. The ribosome then reads the code on messenger RNA. Then the anticodon from tRNA matches the codon to mRNA, and as a result, it's able to bring in the correct amino acid, and then those amino acids bond together to form that peptide bond. Protein is then made once all those amino acids are joined correctly. And so that's what you needed to know for that worksheet. Hopefully that helps you, and you can double check your answers and use this as a way to study for future formative and summative assessments.